All right, our class started, and so there. All right, what are we going to do this week? And we're going to finish reading chapter 16. Chapter 16 is about receptors and signaling pathways. We're going to do the homework based on the almost hour-long video. It's a pretty long video with lots and lots of learning to do throughout it, but a specific focused assignment in it to draw and label it, and send me a pic of your drawing assignment before class next Tuesday. And then we're going to have a quiz on that video, or the whole video, at the end of class on Tuesday. And then you're going to start working on your term paper. And I made a video, just 8 minutes long, that should describe everything you need to know about the term paper. And I'll make a Brightspace announcement with the video, and then I'm going to share with each student these two Google Docs. And the Google Docs are explained in the video. And so we're going to be off running on that. And I'll have a description in the Brightspace announcement too. So we'll start rolling forward on that. It's not due till the end of the semester, but you'll see the Google Docs are there in part for us to work, kind of plug away at it. And we'll talk about it in class next Tuesday, once you've gotten a chance to see what that is. Then 2 weeks from today, we have our first exam. What's our first exam going to be like? It's going to be like 2-point questions. I will send you the first exam from last year, not with the answers, but just with questions so you can see, oh, that's what we're targeting, that kind of detail. And I should have done it by now, and I just spaced it out. So if somebody reminds me in an email, if I don't, just remind me to email, and that's great. So I'll post that, because that's a good resource. And again, it doesn't have the answers, and lots of questions will be, completely different, because it was a different semester, and so we had different stuff coming up like that. But it shows you like what the expectations are, and so you aren't just like, whoa. And remember, one of the things is you kind of have to work away at it, because it's a time test, not that you're going to run out of time, but just that our class meets for a certain length of time, and then another class is in here. And so you have to be kind of direct, like we saw last time when I put up that thing about the mind map, the concept map of the estrogen receptor. We know a lot about it. And if I ask a specific question about the estrogen receptor, you just want to limit your answer to that question and not be like, and I can answer a lot more, right? Because it'll just be like 2 points, 2 points like that. Okay, so we're rolling toward that now. It's still 2 weeks away, all right? And so that's where we're at. If you want to use, I put some markers outside my office for that drawing assignment. I try to do that just to encourage you to draw in color and big markers rather than little tiny pens and one color like that. Yep, that's just a good way to learn, and I'm trying to help with that. And some of that stuff you'll be like, yeah, that's useful for me, and other stuff you won't over time, right? So then the term paper, yeah, and I just wanted to show you that, but I forgot to get I just took a little screenshot of the video I made, because it's a very simple video, but just rather than try to explain it once, right, I can explain a video, and then you'll have that there, and you can check in with that video as you get forward on that, okay? And so this is what we're at, and we have this giant, central big course, big picture, and it goes like this. Chapter 7, we started our course with it. Let's learn about genes and transcription regulation, turning on and off genes in cells, yeah? And so it's all about transcription factors, and they're proteins, and so we are learning a lot about proteins. So then we read this chapter about chapter 16, and it includes that estrogen receptor story. And remember when we started our course off, I said, here's this MyoD story. It's muscle differentiation, and we're going to have the MyoD gene get turned on, and then the MyoD protein transcription factor immediately turns on myogenin, essentially, like that. Myogenin gets made into RNA and protein, and it's a transcription factor, and it kind of immediately starts to activate the muscle and gene expression, yeah? So then we brought up estrogen receptor for a reason, and that is that the estrogen receptor has a built-in delay. So the first story, myodine, myogenin, muscle, goes ding, 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 right in a row. But lots of these transcription factors are regulated, and we've seen a list that we're building, right, of how they're regulated, and that builds a delay and control over the cascade of gene expression that can occur. And the estrogen receptor is a really good example, because in female vertebrates, there's a big delay between embryonic development and adolescence or puberty, when the estrogen receptor plays a big role in sexual differentiation of all vertebrate females. See, and so that's what that chapter is bringing in part, and it's a big part, and that is we'll see something like the PDGF protein signaling molecule going between cells, like going between our platelets and our wound skin cells, and they got to be stimulated. So that's going to go in between, and the signal doesn't come into the cell like it did with the estrogen receptor. It's on the surface. So this signal transduction cascade, and its target often includes proteins like ELK, transcription factors that get switched on. So this presents the idea of ability to really have that blink, 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 have a delay, and all kinds of other features of regulatory orchestration like that, and really bringing the otherwise blink, blink, blink to life, okay? And so then, so what might the cell do, right? So it's delayed that activation. What might the cell do? Well, in the case of estrogen receptor, go through puberty. In the case of the skin cells, go through the cell cycle. So how are they going to do that? And in this case, there's going to be activation downstream of the signaling pathway of MYC. And MYC is a big deal because MYC is an oncogene, so we'll be interested in that. And it's a transcription factor that's going to drive the cell through the cell cycle. That's the next chapter. And we probably won't read any of that for our exam. Maybe we'll look at a couple of figures, but we're actually leaning into it. When we say MYC does something, we'll talk some about what MYC does. Probably not today, but definitely next Tuesday. And so, you might, and we'll talk about it in class next week. Maybe there'll be a little bit of chapter 17 cell cycle stuff that's on the exam, but it'll definitely be kind of leaning into that for our first exam. But those are the topics. We already have a good handle on what's going to be on the first exam. It's everything that we've dealt with, so the quizzes and the assignments we've had, but then everything that's happened in class, and those are the highest priority by far. And then further understanding into the book. First, to really understand what we're talking about in class really well, and practice it, and try to draw stuff over yourself. And then also go further in the book, because there's more details in there, but they're going to be the last thing. There'll be some students that don't do as well on the first exam as they want, and they'll say, how can I do better? And I'll say, emphasize what's happening in class. Get that down really well. And we've seen, like sometimes like on the quizzes and stuff, and we're questions come up in class, it's really important to be able to restate it. Like when you're learning a song, you haven't learned it unless you ever sang it. And you're like, oh, whoops, I didn't learn that many from, I know that song, but you don't, And so that's true for learning too, you gotta do that. So that's where we're heading, and that's the core of our course. Great, that's a good core. So here we are in chapter 16, and we're gonna go through this list of receptors before the first exam. They'll all be on there, this whole chapter definitely will. And we're talking a lot about that one. That's in the video assignment and the quiz next Tuesday. And it fits into that category. And so we're focused on that category right now. It's not the first category that comes up in the chapter, but it's the one we're going to focus on as we roll into it. And that's for sure our favorite receptor signaling pathway by far. We'll learn the most about it. We also learn the most about myogenic, because they're a classic example of all genes and how all genes function. There are always two parts. The same is true here for the PGF signaling pathway. There's always a signal, a receptor, a signal transduction pathway, and then a cellular response. But it might include new gene expression to do stuff. It might include other things, and we'll learn some more about that. And so that's where we are just specifically in the chapter, right? And you can see the chapter lists these out, and they're all equally important, but there are some things that we'll emphasize a lot and some we won't. We learned a lot about the estrogen receptor, and as specifically well in nuclear receptors, we'll learn a lot about the PGF receptor as a specific example of these tyrosine kinases, okay? All right, so these receptor tyrosine kinases. And then I just put this arrow up here, because our book has a figure, and I'll show it today. The book has a figure about a variety of small molecules that are signaling molecules. And books do that, where they're just, hey, take a look at this, right? And we don't need to learn anything about that. So it's really good to bring questions to class about sections in the book and be like, what are we supposed to get out of that section? Because a lot of times it's nothing. For somebody who's learning stuff for the first time, it looks like a really appealing figure because it has a lot of chemistry on it, and you have taken a lot of chemistry. So you're like, I'm good at chemistry, and you see names, and you'll be like, I learned the structure of those molecules and their names. And there's just no chance. Even if you were like getting a AAA+ in this course, there's just no chance that a question about that would show up on this test. Because we have better things to focus on. Just by saying PDGF, that's a signaling molecule, and it's a good classic example. Yep, like that. So That's true. Okay, so this is where we're at. Oh, and there it is, right? I mean, that looks sweet, you know? Go for it. But it's, you know, we're not looking at the chemical structure of those because they're just signaling molecules and they just bind to the surface to a receptor. And then the receptor activates a signal transduction pathway. And we're really interested in those things more. And so there's our pathway, and that's PDGF, that's a PDGF receptor, and RAS is on that pathway. So as a signal transduction pathway, one we're learning that is involved in receptor tyrosine kinases is the RAS signaling pathway. And people talk about RAS, and on that same pathway, they also talk about RAS, MEP, ERK. And we don't care so much about them. They're just clink, clink, clink on that pathway. But that's actually where this pathway gets its name. And so let me just skip back to that. So it says here in the title, it says the signaling by MAP kinase. And so that's actually where people talk about it. Even though RAS is there, they talk about it, they name it that way. So the first pathway we learn is the RAS pathway, or the MAP kinase pathway. And so let's just learn the name of what MAP stands for, just because it's kind of satisfying to learn that. And so it says MAP, and what it's referring to is the RAF, MEC, ERK guys. And we don't need to know much about them. They are MAP kinases. And so the P stands for they're all protein kinases, and so we've seen that. And why are they there? They're there for amplification, because each one's an enzyme. So each one enzyme molecule can go. So one RAF can activate many MEC, and one MEC can activate many ERK. So we can really kick it up. But it's also there because there's this potential for integration. And so with each step, it's a required step. And so sometimes there's some other pathway feedback, either positive or negative feedback, to integrate that, like that, right? Okay? And there's a third one that we'll come up with in general today, but we don't need to mention it yet. So RAF, MEC, ERK. And they all do the same thing. They're protein kinases. So that's what they are, protein kinases. And the M is just kind of the curious word. And the M stands for this word. It says it's mitogen. And a mitogen is a molecule that stimulates mitosis. So it's an old-time word for a growth factor. So PDGF, platelet-derived growth factor, is a growth factor, and it's a mitogen. It stimulates growth. It stimulates mitosis and the cell cycle. And so MAP kinases are mitogen-activated protein kinases. Sweet term. So that's why they get the famous name of this pathway. We're going to learn 3 different signaling pathways today. The first one is the RAS MAP kinase pathway. Okay?
So the A is the amplification.
Yeah, activation. Yep.
Okay.
Yep.
What about
So
the I? That just doesn't play a part.
MAP kinase.
You said like the integration for 2.
Oh, yeah, that's not part of the name. I should say that more clearly. So that list of amplification, integration, and the third one is going to be variety. That's just talking about the principles of the signal transduction pathway in general.
Okay.
The reason there are those signal transduction pathways is because we can amplify the signal and we can integrate it, get feedback loops, and you decide, is this really what we want to do? And then we can also add a lot of variety to the signaling pathways. And that comes up, that variety is coming up today because we're learning, and we'll learn some more about the RAS MAP kinase pathway, but there's a second pathway in here that we're going to learn about too. It's called the PI3 kinase pathway. And then in previous editions of your book, I have the figure, and we'll learn a third pathway just in class. And so they're all receptor tyrosine kinase pathways, and there are more than those, but those are 3 good ones to learn, really famous ones to learn. So, right? So see, estrogen, we've made a concept map about that in another year, but it just goes spraying out. Like when we land on PDGF, it just goes spraying out because it's a receptor tyrosine kinase, and it's our favorite example of that. And then they work by activating signal transduction pathways that are super famous and significant. And we'll focus on the RAS MAP kinase one, but there are these two others that we're going to learn just so we can see variety. So depending on the cell in different situations, in some cases they activate one or the different pathways, and there's a big variety of that in our biology. In other cases, in some cases, they even activate two pathways. So in some situations, the PDF3 receptor activates the RAS kinase, and it also activates that PI3 kinase at the same time. So we get this variety of responses or pathways into the cell for responses. Okay? So just on this, and so we'll loop back to this RAS one a bunch. And so the other two will be just kind of a one-shot time in class. And they're in our book, and we need to know them, but we won't elaborate on them because we really want to elaborate on one signaling pathway to be like, oh my gosh, I see that one really clearly. And it's this one, RAS, RAF, MEC, ERK, ELK, like that, right? And in our pathway, there are a bunch of things that happen in RAS signaling pathways. This is just one, and one is that we could activate ELK, and ELK could activate MEC. ELK is activating other things, but we're just going to pick on MEC so we can have some other elaboration of that pathway. And MEC is a transcription factor that we're going to especially learn probably a little bit after this first exam about the cell cycle, because MYC stimulates the cell cycle. It's a transcription factor that's going to stimulate the skin cells under the scab to heal. So just see this, right? The MYC oncogene and ELK1 are both involved in gene activation and cancer. So they could be mutated. Remember back in the 1970s, scientists found MYC first in one of these oncogenic retroviruses. It's a big time carcinogen oncogene, not carcinogen, oncogene. MYC is a transcription factor that can drive cell proliferation and metabolic reprogramming. ELK is a protein that can activate the c-MYC promoter. And they refer to it here as a promoter, but they mean the regulatory region. This is an example where some people, different people, use some different terms for the parts of a gene. And these people are talking about promoter not the way we talk about it. They talk about the promoter like the entire cis regulatory region. That's fine. We're not going to get surprised by that. We're just saying that ELK must bind to an enhancer of the MYC gene and activate its transcription. And we need to know that, and you'll see we're going to become experts of that, if not today, next Tuesday, about that MYC, because the MYC is sick. So then these proteins, yeah, they're in us, but there are orthologs of MYC, receptor tyrosine kinases, a bunch of them, and MYC, and RAS, and RAF, and MEC, and ERK, and ELK in fruit flies, orthologs. So this is a very ancient signaling pathway in animals, and it's used to stimulate proliferation during embryonic development or juvenile growth or recovery from an injury, like a cut. Yep, big time orthologs like that. And so our favorite example of a receptor tyrosine kinase and RAS and MAP kinase and MYC is healing under a wound and moving the cell cycle forward. But it happens in cancer where it gets disrupted, and we've talked some about how that happens to RAS. It happens to MYC too. And it's cool because the mutations that happen to MYC in cancer are different. Remember, the cancer mutations that happen to RAS are always in the coding region. And we'll look at that more, and it's looked at in that video. They're always in the coding region. You're going to change the product, the protein product gene of the gene, the RAS gene, is going to become defective in this really curious way, where it's going to fail to turn itself off. MYC is a totally different story. When MYC is involved in cancer, the mutations are only in the regulatory region. So they're really good examples of that idea. It's very physical, it's very logical. Here's just some of the general biology of the receptor tyrosine kinase, because the book talks about them all. It's a big family. I don't know, I think there's 60 of them in us. 60 receptor tyrosine kinases. We're going to learn especially about PDF receptors. So it just shows when the growth factor binds or the ligand binds, we get the two monomers together, and it's tyrosine kinase. It autophosphorylates, and now it's activated, and it will bind to another protein or molecule and bring that message into the cell through that signal transduction pathway. There, our book is talking about the RAS and exchanging GDP for GTP to become activated, and then breaking down GTP. This is that protein, the helper protein, that is frustrating because it's helping RAS, and so you can be distracted by it. But remember that we know where the problem is with RAS. It's in the RAS protein in the active site where it's failing. So even though it needs help to do that enzymatic activity, it's just helping, it's not doing it. So our big focus is on RAS. It's really an advanced topic to talk about how other proteins are helping RAS get the job done. That's fine. It just brings them into the cancer story. If you say, oh, you have a problem with that GAP protein, well, that's going to help keep things going too. So, it's an advanced topic that we don't have to worry about. So we've shown that before, that we'll learn about those oncogenes, oncogenes that were originally studied in the 1970s as oncogenic retroviruses, like that. And there's RAS, and remember, we're remembering that it's a monomeric G protein, because we're going to meet another kind of G protein in this chapter, and we ought to keep them distinguished. So let's just read through this quickly, because it's just people just crank on RAS, right? They've known about it for 50 years, and it's the most common gene mutated in human cancer. So there's so much research done on it. So I'll just read these highlights. So RTK RAS signaling, so that's receptor tyrosine kinase RAS signaling pathway, is the most frequently altered oncogenic network in cancer, with 46% of all samples displaying alterations. They're including mutations in RAV or anything else in the pathway, MYC, like that. So almost all of human cancers are what we're talking about. So good choice. Good choice because it's so important also, not just in cancer, but our normal health, and then also because it's such a good thing to learn as an example. Introduce this. So it says that the pathway, and then we'll just kind of scroll forward some of these highlighted sentences I have. So RAS, actual mutations in the RAS gene, contributing to almost a third of all human cancers, people will say. Then they're saying KRAS, and I mentioned before that the RAS gene is a member of a 3-gene family, and we do not care about that. So that's why they're saying KRAS, Kirsten RAS, don't worry about it, just RAS for us, right? So they're just essentially saying that, but here in the one time we'll look, because the different RAS genes have different roles in very specific cancers of people, like that. So KRAS mutations are exceedingly common in pancreatic adenocarcinomas and colorectal cancers. So they just said something, right, that we're just starting to introduce to. And we got to introduce that in the estrogen receptor, that types of cancers, breast cancers, it's a specific kind of cancer, and then within that breast cancer, it's a specific kind of type. Is that ER positive or ER negative? And they're saying the same thing here, that for specific cancers, there's specific oncogenes that have specific mutations that are really common. So overall, you go, wow, RAS is significant in cancer. Almost a third of human cancers have RAS mutation. But more specifically, there are some kinds of cancer, specific types of cancer, where they almost always have a mutation in RAS. And what that's saying is that tissue, when it's normal, it already is using RAS in signaling. And so they're talking about things here that tend to be like gastrointestinal type cancers. And our cells there have to replenish like they do in our skin. So RAS is playing a role, it's playing a normal role there, and that opens a door. In other cases, like maybe I'll just make it up, maybe you never see a RAS mutation in brain cancer, right? A third overall, total, but maybe never in brain. And it's because the RAS signaling pathway just isn't used in brain. RAS is about growth, and the brain isn't doing any growth. So maybe it's something else that's common. And you just have to do it on a case-by-case basis. And doctors and scientists are doing better and better at looking at what's going on with this patient and their specific mutations we want to try to treat them, right? So KRAS are like, well, NRAS, another RAS gene, more common in melanomas, thyroid cancers, and leukemias. So see, they fall in this different way. And so we're not going to find a silver bullet, we just cured all cancers with this strategy. We're going to try to take them on one at a time, which is a pretty bold effort, right? So although KRAS, HRAS, and NRAS, the 3 RAS genes, share functional similarity, then we get this very specific fact. KRAS missense gain-of-function mutations. So those are the mutations in the codons, or the coding region of that KRAS gene. They tend to occur at the 12th codon, and we talked about that last time. Very specific, that's in the active site. So you're disrupting the active site and damaging it. Well, those HRAS and NRAS mutations occur in the 61st codon. So a different position, but same effect on the active site. Differentially used across cancer types, blah, blah, blah. So you see different frequencies in different cancer types like that. Super interesting and significant details. And going back to, yeah, but in general, for our course, it's in the coding region. And we know why. It's because it's got to turn itself off. And if it doesn't, that's going to lead to cancer. And the most likely mutation you have when they're random is it's not going to do its job as well. And it doesn't do its job as well. Something like that. Yep. Okay, great. So lots and lots of interest in RAS. And there's RAS up there, and you can see it's floating around and it binds to RAF when it is activated. So that's how this signal moves on. It's when RAS is activated by having GTP, all it does is it binds and it holds RAF and activates its mitogen activated protein kinase activity. Then you see, phosphorylated ALK, and we talked about that. That's a good example of that. So it's a really great pathway to talk. So now we're kind of done with talking about RAS and PDGF receptor, but you're learning a lot about it in the video and the assignment and the quiz. So we'll refer to it through the rest of our course. Yeah? But we have a nice core. So now we're just going back to, here's just some general pictures from the chapter. So the chapter starts out introducing the idea of signaling. And we can just draw that out in a general way here. And it would look like this, where you say, when we talk about signaling, there's always going to be a cell or a group of cells that we can call the source cell. And they're going to make a source molecule. And it could be a hormone or some other molecule, and it could be a protein or lots of other different small molecules. And that is going to bind to a receptor, and we could say a receptor protein, because they're always proteins. The signaling molecule could be a protein like PDGF, or lots of other chemistry, steroid hormone, blah, blah, blah. And the receptor is either going to be in the cell membrane, that's typical, or it'll be a nuclear receptor. And then there's going to be a signal transduction pathway. And I forgot to put a title up here. This cell is generally called the target cell. So we have a source cell that makes and releases this signal, and then a target cell that responds. And so it's going to have a signaling pathway, a signal transduction pathway, and then it's going to have a cell response. It's going to do something. And one thing we like to talk about is it might activate gene expression, might express cells, and might express new genes. But it doesn't have to, and we'll learn a couple examples of that in this chapter. So that's just what the book starts out with, trying to give you that overview of this, and then it makes the mistake of showing you these detailed figures of structures and names we don't need to know. So those are examples of some of the ligands for nuclear receptors, right? We don't need to know the structure of estrogen or any other of these. We do need to know the word estrogen. It's the ligand of one of those. And so we've learned a lot about that. And that's one of the first sections in chapter 16. And it cranks back through the stuff we learned, so we really want to know that well. And it probably adds a little bit of knowledge to it. But we don't need to learn much more from the book about that. We didn't need to know the names of the other receptors. We are learning about the insulin receptor some. We don't need to learn the structures of the ligands and the names of the ligands like that, except estrogen. And so there's a figure from this chapter that we talked about. There's the estrogen receptor working as a transcription factor and a ligand-dependent transcription factor. It has a DNA binding domain, it has a ligand binding domain, it has a transaction domain. So we know so much about it. And so it's there, and there, and there. And then similarly, we're learning a bunch about this pathway. And so that's the receptor tyrosine kinase pathway. So we talked about the estrogen receptor, and we've talked about the PDGF receptor a lot. And so now we're going to talk about them in general. And one thing is this: amplification, integration, and variety. The same receptor can activate different pathways in different cells or multiple pathways in the same cell. So now we're going to learn more, and we're going to do that thing where we've learned for receptor tyrosine kinase, one of the common signal transduction pathways is the RAS MAP kinase pathway. So here comes that second one, okay? And so we will learn about 3 receptor tyrosine kinase signal transduction pathways. 1, the RAS MAP kinase pathway. 2, the PI3 kinase pathway. And 3, the phospholipase C pathway. The first 2 are in your book, the third one is not, okay? And what the book is just showing you is that variety notion, that these are receptor tyrosine kinases and they'll activate a response pathway, but it's not always RAS. It could be one of these other ones. And there are additional ones, but these are big, famous ones that are often used. So the first one, RAS is that pathway. So we are, that's our fave by far. And just like for today, we're going to learn these other 2. We're not going to go back to them a lot. Yeah? So then the second one is a good one, famous one, and we've actually seen it since day 1. It's the signal transduction pathway that's shown on that FOXO slide. So we already have seen that. So there's going to be a growth factor and a receptor and then a signal transduction pathway, and its target is FOXO. So we're going to go to that. So that's one I showed on the first day, and we've gone back to it because it's so cool, right? It's so cute. That FOXO is a transcription factor, and it needs to get into the nucleus to do its job. And it's such a great example of the physicality of molecular and cellular biology, right? Like imagine you're talking to your car mechanic, right? And you've like, I've got a can of gas. And they're like, yeah, it needs to get into the car to do anything. And you're like, well, yeah, but I have it. And they're like, yeah, no, sorry. You had to put it in the car like that. Like this is my favorite advertisement of all time is a radio ad. So it's a radio ad, right? And it's a person who's calling in. I don't remember what's advertised, but it's a person who's calling in the help desk, and they have bought a new computer, and they're at home, and it's not working. And so it's 2 voices, the consumer, and then this computer help desk, right, person. And they're going through these questions one at a time. And the last question in the ad is, the help desk person says, do you have the computer plugged into the wall? They've gone through everything else. Do you have it plugged into the wall? And the consumer says, this computer didn't come with a wall. And you're just like, whoa, yeah, like that, And so, right, it's physical like that. It's physical like that where you're just like, woof, It's physical like that where you got to put the gas in the car. Or FOXO has to get into the nucleus, and you have to be gentle about it and be like, it's cool and everything's cool and groovy and far out, but, we got to get FOXO in. So it's such a good example, right? But that's what we're going to look at closely, because that's actually PI3 kinase. So from the big view, see that is going to be a growth factor, some ligand, and it's binding, and this is a receptor tyrosine kinase. And we aren't necessarily going to learn its name, but that is actually the insulin receptor. And you know what insulin does, and this is a role for insulin that's very different than what you know. But that's the insulin receptor, and so we're going to learn that one, because we're going to learn the names of 2. And we learned 2 because the PGF receptor and the RAS, MAP, RAF, MAC, ERK pathway, and MYC, that's such a good one. And the insulin's a really good one too, and we don't worry about the details of it being insulin. We just want to get to this figure, because it's going to activate a very different pathway. And so all we have to do is zoom in on this and look at that carefully. So the receptor gets activated, phosphorylated, and it is going to activate a pathway that includes that. So that's the name of the pathway. It's a PI3 kinase. And kinases, we know, they phosphorylate target molecules. So this one phosphorylates a phospholipid in the cell membrane. So it's shown there, and it's a big surprise that it's not a phosphorylating a protein; it's phosphorylating a target phospholipid, and we can learn the name of it, and the name is PIP. It has a long name: phosphatidylinositol triphosphate. PIP is great for us, right? The structure we don't need to know. We need to know that it's in the cell membrane because it has a phospholipid. It has that hydrophobic tails and hydrophilic head. So it's there. And what's going to happen is all this is going to do is it's going to phosphorylate. It already has some phosphate. It's going to add another one. And that activates it. And then this is going to activate mTOR. We don't need to remember that. But you'll see this AKT, it's a different kinase, right? It's not RAF, MAC, ERK, it's a different thing. And we'll eventually learn that AKT, because it comes up in later chapters. So just so you know, I probably won't come up in the first exam. This doesn't come up very much in what we're covering yet. But we'll learn about that. And look, it phosphorylates FOXO. So see, it's a nice pathway, because we think activation of receptor tyrosine kinase, PI3 kinase, something, AKT phosphorylates FOXO, golden. Yeah, that's how that one works. It's great. What we're going to do now is we're just going to look at that phospholipid molecule some, because it's a freaky thing to see that. Okay? All right, it's just in the membrane. And so here's the deal. Lipids have three main roles. The one you think about is energy storage. They have a lot of calories per molecule, right? But then you know it plays a structural role too, because you know that lipids make up this, the phospholipid bilayer. And this is the third one, and it is that it's involved in signaling. For example, estrogen is a lipid. So it's involved in signaling in a lot of different kinds of molecules. It's involved in signaling here. A phospholipid in our cell membrane called PIP is involved in the structure of the membrane, but also in a signaling pathway. And again, we don't need to learn the structure. We skipped over the chapter that the structure is in. This chapter 2 in our book is about some chemistry, right? And we're not going to learn a ton of chemistry here like that. You've learned a bunch. But it's a very familiar looking thing. And it's phosphatidylinositol, and then it will be, have some phosphates that have been added, like that, okay? So they'll refer to it as PIP2 and PIP3, and we don't need to dive into the details of that. Just PIP would be a great word to remember, and nobody needs to remember more. So the PI3 kinase, that's labeling, that's naming what it's going to phosphorylate. It's going to phosphorylate PI3, it's just using to say PIP, and PIP3 is what it's really saying there in its name. So that's its job, and we just want to learn that one, right? Because it's different than RAS. It's just not the same. But there's reason to be excited about it because there's amplification, there's potential for integration, and then this variety, right? And so we're almost already done talking about that one, PI3 kinase. Great. So then here's this phospholipase 3C pathway, and it's no longer in your book. So now for the next 5 minutes, you'll just learn everything we need to learn for our course for this phospholipase C. It's the third one, right? The first thing is, remember, we're still talking about receptor tyrosine kinases. So there's a big family of them. Our favorite is the PGF receptor. We learned a little bit about the insulin receptor, but not very much. And in that big family, they can activate different signal transduction pathways. Three of the most famous ones are these three. Yep. Okay, so the phospholipase C pathway. And so, again, no longer in your book, but the reason we're talking about it is because it activates three super famous second messengers. And there's a chance that way back in intro biology, you remember the word second messenger. And these are famous second messengers, and second messengers are on signal transduction pathways. And they're called second messengers because the first messenger stays outside the cell. And so that's like the first or primary messenger, and nobody calls it that, but that's what they're referring to when they say second messengers. Second messengers are inside the cell. And we're going to learn the names of three: DAG, IP3, and calcium in this one slide. And so I zoomed in on this, and you can see what's happening here. It's really similar looking to the second one because that PIP is involved, but it's different. The previous one was a PI3 kinase. So what did we do to PIP? We added a phosphate. This one is a phospholipase C, and it cuts the PIP molecule into two. And when it cuts it into two, it makes two secondary messengers. So the PIP is there, the secondary messengers are not. But when phospholipase C cuts it, they're there. And one of them stays in the membrane because it has the tail, and it's DAG. And it just floats around, but it's active now. It's a second messenger, and it's like, go. And this is what it activates, and you just have to learn that. that activated DAG for us activates this protein kinase C. And then it's doing something down a signaling pathway. And then completely separate from that, this IP3 is the other part of PIP. And it doesn't have the tails, so now it's free to float away. And it floats around in the cytoplasm, and it eventually activates receptors inside the cell as a second messenger. And the most famous thing it does is it releases calcium. So those are our three signaling factors we get out of this. Now, you know, you might remember second messengers like that. Now, there are, in the RAS MAP kinase pathway, there are no second messengers. That's fine. People love to talk about second messengers because they're exciting, because they play so many different roles. But some signal transduction pathways have them, like this one, and others just don't. So that's why we picked up this third one, right? The odds are dropped from this the chapter because it's just there's a lot of stuff in the chapter. When you look back at earlier editions, they've just been cutting, because it's it's the center of our course, right? And every time they cut a figure, I'm like, too bad, you know. And I write them a letter, and I'm like, too bad, you guys. You know, I'm not gonna, maybe won't use this book again. And they'll be like, come on, Dave. Yeah, you know, like that. But yeah, so see them, there they are. Phospholipase C gets activated when the receptor gets activated for the ligand, and it cuts that PIP into two parts, DAG and IP3, and then IP3 floats in and really releases calcium. They do other things. This is part of the variety, not just three different signaling pathways into the cell for receptor tyrosine kinases, but this pathway alone opens up a lot of different variety of opportunities for how the cell could be getting that signal transducted into it.
Dave,
So, yeah.
why do we not consider the kinases of the other pathways to be second messengers? There's
Yeah,
so
because
many
second
cells.
messengers is a really kind of just a loose, casual term, and it's referring to small molecules rather than proteins. Yeah. Yeah, and so it's just, but again, you know, you see it because they want to trot that out, because they want to trot out the idea of there's stuff happening in this cell. Yeah, and so the first one, there's DAG, that's the one, and it has a longer name, and we don't need to learn it, but I'm going to show it to you in a second. So there's the first one, right, DAG, and it stays in the membrane and it activates that protein kinase C, right? And so that's actually not really its full name. Its full name is right here. Everyone calls it diacylglycerol, DAG. Don't worry about it. Diacylglycerol, DAG. Maybe you've heard it before, you know, because like nobody calls it DAG. They call it diacylglycerol. But don't, you know, that's just, you know, and you can learn that, but don't worry about it, you know. Then here's the second one, IP3. We talked about it. What does it do? It gets cut, and it's the head of the phospholipid. It's very hydrophilic, so it floats off into the cytoplasm, and it becomes a second messenger, a small molecule that signals, and it binds to a receptor that releases calcium into the cytoplasm. And there's the name of that. Again, we can just learn PIP and IP3. Great. So PIP gets cut into DAG and IP3, inositol triphosphate. Another one that nobody ever calls IP3, hardly. You call it in some places, but it's unusual. People just call it that. But we don't need to worry. I'm just showing it to you. Yes?
Can you say PIP gets cut into the DAG and the IP3? That is what, okay.
Yep, and it gets cut by that enzyme, phospholipase C. So when phospholipase C gets activated, it cuts PIP into DAG and IP3. And then
Where is PIP on that diagram?
it's right here.
That, okay, that
Yeah.
PIP2, okay.
Yeah, and so it's showing it catalyzing that chemical reaction. That's a single step chemical reaction with an enzyme, a substrate, PIP2, and product. Yep.
So the calcium's not inside the cytoplasm at all till the IP3 binds.
Yeah, so with regard to calcium, calcium is a really famous inside the cell second messenger. Like it plays a big role in muscle contraction as a second messenger. When the neurotransmitter arrives and to contract, calcium really spreads that. And famously, what happens to calcium in the cytoplasm is it gets pumped into some organelles. Like this is probably the endoplasmic reticulum. And they refer to that as internal stores of calcium. So the cell is intentionally hiding it in organelles, and then this receptor can be activated and release it, so we can have a burst of calcium just floating throughout the cytoplasm outside the organelle, but just floating around inside the cytoplasm. Yeah, so that's what's happening there. Yep. Yep, and that's really standard stuff. And so then the third one is this calcium, and I'll just show you this. Again, I don't think it's in our textbook anymore. Maybe it is still there. But this just shows that it's really not calcium. Calcium is always bound to a protein called calmodulin and is activating calmodulin. No big deal. But just another protein that is a protein of big significance on these pathways. Okay? And so, again, another thing that we don't need to worry about as much, and that is these signaling molecules include neurotransmitters like that. So, anyway, that's all fine. So we introduce these second messengers. And I think it's a really nice way to see these three, and I hopefully have the three now listed again. Maybe not. We'll scroll back to them. The three. Boom, 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 right? You can take your learning and memory to the three different pathways. The first one we're doing for a homework assignment. You're going to take a picture of it and send it to me. Then the second one we've been looking at, it's FOXO. And we want to look more carefully at it and add the PI3 kinase story and name it that. But it also has that activation of AKT. AKT is the kinase that actually phosphorylates FOXO. Great. Now we have this third one, and the third one's memorable because it has these three really famous second messengers in it. So, you know, that's why you pick them. There happen to be prominent, really important ones, but they also are coolly distinct from each other. Yep? Yep, so we're doing great on that. And then, so we have here, so we're talking about that, and we're just, you know, deep diving into receptor tyrosine kinases today. And and then here's a third one, and this is, so we think of PDGF receptor, yeah, that's that RAS MAP kinase one. And then we think of the insulin one, and then we didn't name a receptor for the third one, the phospholipase C. And that's intentional because one of the reasons we're naming them is to name their variety. And we don't want to do too much effort to say, yeah, you know, PGF always goes with the RAS, okay? Because there's flexibility, and so I'll just show you this, right? So several receptor tyrosine kinases can activate phospholipase C. Like who? Well, like PDGF can do it. So under certain situations, our favorite one, skin healing, the famous pathway is that PDGF receptor activates the RAS MAP kinase pathway. But in the other situation, it might activate that phospholipase C pathway, like that. And same with the insulin receptor. So do we have an example of the receptor for the third one? Yeah, well, weirdly, it's the first two, the PGF receptor and the insulin receptor, because that is that variety, you know? So we don't need to, you know, get too fond of always thinking, oh, PGF must only work through RAS. It can work through this other one, like that. So variety, right? So you just want to be that, and this is just this general thing. Signal transduction pathways are there for amplification, integration, and variety. Sam is going to come back next Tuesday and talk about that CAR, chimeric antigen receptor. It's a receptor, and it's going to activate 3 different pathways in one receptor. That's how they make a super protein, is they get it to do all 3. So we'll learn about that and Tuesday. And it's just a good example for us to visit, you know, 4 times during the semester. So now we're going to talk, are we going to talk a little bit about these? Maybe. I think what we'll do is, I think I'm going to just jump ahead as we talk about the receptor tyrosine kinases, and we'll move to MYC and talk some about MYC. And then we'll go to these G protein coupled receptors. The G protein coupled receptors are a big, big story, but not as big for us because they're not so often involved in cancer pathways. They're involved in, you know, health and disease, but not so much cancer. And so I think, and I've got these slides just a little bit ahead, and I think we'll do as a kind of an early concept chat, and then we will, you know, so we won't really transition to something completely different, but we'll do kind of a deeper dive into looking at MYC. Because MYC is just totally messed up as an example of an oncogene that we should learn about in our course. Okay? So let's do a little concept chat. So again, get up, walk around, be like, oh, I haven't talked to Lydia because Lydia's way back in the corner. You know? Like that. Okay? Yeah, so, and try to make 2 people. If you look around and you have 3, go like, okay, maybe there's an uneven number of people. All right? So what are 3 important things that we talked about today? And hopefully you'll be referring to your notes and your neighbor's notes and working it out. Uh-oh.
Yeah, I learned about how the math name came about because I was watching lectures, so I was watching the video last night, and it kept saying like the name now. I'm like, this acronym does not work. So that's nice to know. So it's
That was a weird.
it's
That's weird how it's put together.
it's like so F is the nitrogen, A is the amplification. Wait, is it activation or amplification?
You say amplification.
Is A amplification or activation?
For what?
For math.
Mitogen activated protein kinase. Yeah.
Thank you.
Wait, what is
Okay.
DHE?
Well, nice catch. Yes.
But I think the second one, I think is a transcription factor.
So ELK is what ELK is what activates MYC. Correct? I mean, that's correct. ELK activates MYC. Because ELK is actually like in the transcription unit, and that's what starts to transcribe. Well, MYC is something to do with stimulating cell growth as well. So is ELK. So I think ELK and MYC work together. I do believe. But I'm going to be rewatching this one lecture. I've not been able to keep up with my notes. I've given up. I'm just going to rewatch it. I also didn't understand the signaling thing. What do you mean by source? Where are these signaling coming from? Very
He was talking
confused.
about different so it's going to be troubling.
Okay. Be like a thyroid hormone, it's gonna come with like a thyroid, and it's gonna be like, you know, like okay. I don't understand calcium yet. I need to rewatch it.
Well, the calcium was just the secondary immune system. Like, if really the purpose of it is important, I think it should.
Is that all like stabilized?
And we'll just do this for another minute.
Is that all stabilized?
It's used in like concentration gradients and seeing.
Yeah, so make it active by all the rest. And we have an exam in 2 weeks on Thursday. I knew that, but I had been repressing it.
So the three purposes of
Okay, Alex, what is one thing that your group talked about that you thought was important so far this morning?
Just like the 3 different ITK signal transduction pathways and how the molecules communicate with each other.
Mm-hmm. Yep, awesome. All right, and then Anna, what's one thing your group talked about?
About like how the MAP kinase is acronym, because I hadn't been able to make that make sense before. So that is the nitrogen, which is stimulates mitosis. The A is an activator, and P is a protein kinase.
Yep, so MAP kinases. And when they were first discovered, they weren't calling them RAF, MEK, ERK. There were other protein kinases they were discovering and stuff like that. They called them MAP-P for the first one, and MAP2P for the second one, and MAP3P. Because they're doing, they kind of, I just mentioned that, you don't have to learn that term, but they're there to just crank it up, amplify it, then integrate it, crosstalk between these signaling pathways, and then this variety. Sometimes they're not even in the cell, right? Right? So like, if we used PDGF, it doesn't stimulate cell cycle in every cell in our body, just some cells. And that's because they don't even have the receptor, or they don't have the RAF or RAS or something, and so you get this tremendous variety from a relatively small number of pathways. Yep. And let's see, and then Lilith, what's one other thing?
We talked about how the purpose or the key parts of signal transduction are amplification, integration, and variety,
Yep.
and how the same receptor can activate multiple pathways.
Yep, yeah, awesome, yeah. And again, it's that physical thing, right, where it's like, well, this computer didn't come with a wall, you know, which is it's just like, oh my goodness, you know, you're just like, that is so epic. But anyway, yeah, so questions people have. And I love teaching this because I love it, but I also love teaching it as a teacher because it's accessible, it's learnable material. It's just a lot of it, you know. So I think it'd be good for any person to take this course in college and just try to learn as much as they could learn about stuff, because you could learn the pieces and they fit into different ways in different places like that. But anytime you have questions, you know, just interrupt, because you're distracted if you're thinking about something and you just interrupt like that. And there are, you know, just even if it's like, you know, could we get that, can we close the door, you know, stuff like that. Yep. All right, so here's some stuff about MYC. So MYC is a transcription factor, and there's a deal with MYC. MYC can get involved in cancer. And so MYC is involved in stimulating growth. And MYC is a transcription factor that's going to bind to the regulatory region of target genes that are needed for the cell cycle. And we can already draw that out just to see, because you'll be like, okay, golden. So again, you know, we probably won't read chapter 17. You know, we're not going to read, you know, chapter 7 realistically. There might be some figure or two I ask you to look at. But chapter 17 is about the cell cycle. And you know, what we add about the cell cycle in our class, you have to learn for the exam. And so, you know, this has this cell cycle is G1, S, G2, and M. And we can just draw a little kind of pretend map about this where this is representing, it's a skin cell. And the skin cells are always growing and replenishing. There are stem cells in our skin that are growing, dividing, replenishing slowly, like that. And so if you looked at a population of 100 skin cells, they would be at different positions throughout the cell cycle. So some of them are advancing through the cell cycle. So you'd see there's a couple in S and then a couple here, and they're just randomly at any given time. Maybe there's only one in M phase. When you look though, almost all of them are here at this point. They're at a stoplight that is the big focus, and people call it start, because it's like a stoplight where they're waiting, and if they get a green light, they'll go through the full cell cycle, like that. And so for this cell over here in mitosis, it was the first one to see a green light, and then these guys saw a green light, and they're just at different places because they're like race cars. They got a different time to start racing around the track. And so you could see, you can count them, and in the video, we actually learn about something called the mitotic index in the PDGF video. And so we'll learn about that. And that's just an indirect way to recognize how many cells are really going around the track versus how many cells are parked. And what we're really interested in is how many cells are parked. If the mitotic index goes up, it means that fewer cells are parked. They're getting a green light more often. And the green light and the red light is a protein that we'll learn a lot about that's called CDC. And it's it's a family of them, and they number them, but we'll really CDK. And that stands for cyclin dependent kinase. And so CDKs are needed for the light turn green. And then when the light is green, that's what it means, is that enzyme is activated, and it's a kinase. So it's choo, 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 and we'll learn about its targets, because it has just fantastic targets, you know, like that, right? Okay? And so start. And it's cyclin dependent, okay? So then cyclins are proteins that bind to the CDK and activate it. So there's a cyclin protein. It's cyclin dependent kinase, so it needs it. So cyclin, it's protein, like that, okay? And sometimes the rate limiting step, and the reason the light is red, is because there's not any cyclin. And that's really what's happening in our skin cells, is they park at the red light for a while because there's not enough cyclin for them to go forward. And if we increase the cyclin, they'd go forward more often. And so now let's just draw a picture of the cyclin gene. So that's a cyclin gene, right? Because it's protein. So if you don't have enough of it, it's protein. You must need to make more, need to activate expression of the cyclin gene. And so there's where this smoking MYC transcription factor is sitting. MYC is a protein, like MyoD was, right? It's a transcription factor. It's got target genes, and one we'll focus on in our course, it's got a bunch, is cyclin. So if you activate MYC, you get more cyclin. You activate CDK, the light turns green more often. See that pathway? Click, click, click. Yeah? So that's what we're doing. And so now you can think about how you could go all the way through it, right? When we get a cut, the platelets in our blood release clotting factor to help clot the blood, stop the bleeding, and make a scab. The platelets also have PDGF, platelet-derived growth factor, that gets released and is going to stimulate the skin cells to heal underneath the scab. That's the only place it gets released by the platelets, just under the scab. And they do that by binding to the surface receptor, PDGF receptor, that our skin cells always have. They're always ready to respond if they get, a cut nearby. So PDGF binds to PDGF receptor, right? PDGF receptor autophosphorylates, activates, then it binds to that other protein that we're not too worried about and activates RAF. So RAF exchanges GDP for GTP and gets going, binds to RAF. As long as it's activated, bound to GTP, it's activating RAF, and RAF is like MAP kinase, mitogen activated protein kinase. So RAF is a kinase, boom, 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 boom. RAF, MEC, ERK, ELK, phosphorylated ELK binds to the regulatory region of MYC and increases its expression. When we increase the expression of MYC, MYC binds to the regulatory region of the cyclin gene, activates it. More cyclin protein binds to cyclin-dependent kinase. That kinase phosphorylates targets and turns the light green. And they're usually not that simple, but we actually have a list of steps for that one example. Yep. There's a woman that has cancer in the skin, a type of skin cancer in the video, and her problem is she's making PDGF in her skin. So everything's getting activated constantly. In her tumor cells in her skin, she constantly has activated MYC that's just going go, go, go. So MYC is another example of sort of a vulnerable situation. If we make MYC, it's going to say go. It's going to say, let's get that light green. Now, in some cells, it doesn't matter, because in some cells, the cyclin is not the rate-limiting step, but it often is. And MYC can jump it over that hurdle, just like that, okay? So what about that MYC? And now we're just going to look at the MYC gene, right? Because it's transcription factor, and it's got to get made. The ELK is going to be activating MYC, right? So MYC is a global transcription factor contributing to various cellular processes, one of which is hematopoiesis. In the bone marrow of adults, 300 million cells are produced every minute. That seems like a ridiculously false number, right? It's a lot. And that's a place where MYC is normally used, is to stimulate our bone marrow cells to make more blood cells all the time. Huge numbers all the time. And we're foreshadowing right now because MYC comes up in some blood cell cancers. That's where it's normally being used, and that means its pathway could get disrupted. So we're going to see examples of that. And blood cell cancers are called like lymphomas and leukemias, and MYC is involved in those in some specific cases. Yes, Alex?
Is that why they do like the bone marrow transfusion things for people that have blood cancers?
Yes, and so there's a situation where they'll do a bone marrow transplant with someone who has cancer. And it's usually someone who has a blood cancer, and that means there's some problem with their production of blood cells and the bone marrow stem cells. And they'll say, we're going to get rid of those, and we're going to replace them like that. We're going to try to get rid of that mutation. It's kind of like doing a surgery to remove the tumor, but you're not going to remove a tumor because it's in the blood. And instead, you're going to try to get another source of fresh and healthy bone marrow stem cells from a donor and just destroy all of them in the patient because there is a mutation that leads to cancer like that, and replace them with these new blood cells. Okay, so we're just focusing in on now the nuts and bolts, like MYC, like MyoD, like that. What's going on with MYC? Okay, and so watch how the body regulates this transcription factor. Remember how it regulates FOXO? Remember how it regulates the estrogen receptor, right? Remember how it regulates heat shock transcription factor? How is MYC regulated? And it's just sick, okay? So it says it's a global, and that means it's used for more than just cell signaling, cell proliferation. But this is the classic role for MYC. So it says the RAS-RAF signaling cascade has an important role in the regulation of the MYC promoter. Again, I mean the regulatory region. Small GTB binding protein RAS promotes MYC expression by inducing the RAF-MAP kinase pathway. Platelet-derived growth factor receptors and SRC kinase also can augment the activity of RAS proto-oncogene. Great, so that's stuff we're familiar with. They're not talking about all cancers, they're talking about certain kinds. That pathway they're talking about, what we learned about. So there's another thing in this, over on this paper, uncontrolled MYC expression observed in human leukemias and lymphomas. Not surprising because it has a normal role in the bone marrow stem cells, okay? Then another expression, the MYC expression pattern is tightly regulated in normal conditions, though MYC is often dysregulated in cancer. That makes sense. Okay, so how is it regulated? Okay, and so MYC expression is highly controlled at several levels. And this is this example where you're like, you have a gun at home with kids, you know, it's not enough to just say, well, we put safety on. If you have like 12-year-old kids, you have to put the gun in a cabinet, unloaded, safety on, bullets hidden, locked somewhere else, the gun itself locked in the gun cabinet, and the key can't find him, right? So that's MYC. It's like, watch out, you know? We have to have it under tight control. So tight control, including transcription, initiation, and elongation. So just to get it transcribed is hard. So when ELK activates, it's really hard. The rate limiting steps for MYC gene expression are really hard to get over. For example, we talked about arrested polymerase. MYC has arrested polymerases stuck in about 6 different places. You know, it's like, don't make this, you know? Like when your brother says, I found a key, let's see what it opens. You're like, don't do it, you know? Don't do it, Doug, you're gonna shoot me in the head, you know? You know, and he's like, I think it's a key to the candy cabinet, and you're like, don't do it, Doug. Yes, Anna?
My question is, why is cyclin the rate limiting step, not MYC?
Well, that's a good question, where you can say that now we're diving deeper further into the pathway, and you could say the actual true limiting step is something that's the one that you're going to regulate, like that's the rate limiting step. So that's a good question, and that is, it's a little bit hard to explain, but like imagine the pickle lady is the slowest person in the, in the booth, right? So you can speed things up by talking to her. You can actually, and we didn't mention this before, but it's an add-on to rate limiting step. You can actually slow the pathway down either by slowing her down or by slowing someone else down below her rate. So suddenly the rate limiting step is the guy making the burger, and the pickle lady is, you know, waiting. Yeah, so that, you know, so yeah. There's one of them is the rate limiting step, you know, but it just takes a little while to think about, well, what is really the rate limiting step in the overall show here, like that. Yep, but back to this. So first one is it's just hard to get it transcribed. Yep, it just does not, you know, we don't want to make it. So that's one, right? A new one, and I don't think I have exactly the full list here because I think I forgot HSF, but you know, we're making a list, right? And it's a new one, is you are not going to transcribe this. Not just the usual arrested polymerase, but that could fit here too. You know, that's an arrested polymerase story, but it's just massive transcription regulation breaks. And people call that transcription elongation. Getting the RNA polymerase all the way down the length of the transcription unit is very hard to do with MYC. So here's the next one. that RNA that you make, right? You make it, and if you're making it, it's just really unstable. So that's the output. We're transcribing it into RNA. It's mRNA because it's a protein coding gene, and it just destroyed almost immediately.
This
So
is the MIC, like if you're
yeah.
transcribing the MIC gene.
if you can get that sucker transcribed, the RNA is going to get destroyed. It does not want to make it. It's just back to that gun example. Go ahead, find the key, Doug, but the gun's unloaded. You know, Doug has got to solve a lot of problems before he gets to that, right? So another problem is it's really unstable, and the elongation can get regulated. It can get increased. The stability can be adjusted, more or less stable. So we have to add that. And this is just MIC. MIC fits into many examples. How are we going to regulate a transcription factor? There are a variety of transcription factors that are regulated by the stability of the mRNA, but MIC is one. It's probably the most famous one. Then translation. If the mRNA hangs in there, ribosomes don't like it. They're like, yeah, no, thank you. I don't translate you. It's really hard to get it translated. That also is regulated. So our big focus is on that transcription regulation, right? If we can activate ELK, we'll make it. But downstream of that, there are other steps, boom, 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 where MIC is being carefully shut down, right? And it's important to appreciate that because we're going to get to a thing where what we're going to do is when MIC becomes cancerous, we're just going to make too much. We're just going to force the expression and overwhelm all these regulatory steps like that, yeah? But that's another step is you really don't want to translate it. Then the next step is if you can make the MIC protein, it only lasts a few minutes. I think I've seen the half-life of how long MIC protein lasts in a cell is 5 minutes. Just don't want to make it, right?
Seems like a huge energy waste to make it, to then not transcribe it, not translate it, and then have it go bad so quickly.
Yeah, that's Doug talk, hey Dad, thanks for coming home from work, but do you think you really need to do all those lockup steps for the gun? Because it just, it seems like a lot,
Well, yeah,
right?
there's a burglar, you gotta like.
Seems inefficient, Dad. You could just leave that stuff open, and Debbie and I get along fine, like that. So yeah, there are a lot of steps, right? But you have to have them. You just like, don't do it like that. It's just a bunch of steps like that, right? Because it's just so dangerous. And protein stability is another one. It's modulated, it's regulated, it's adjusted, right? It's like, get it, or don't get it as much, right? And again, we're focused on that one, because it turns out just the transcription regulation with the transcription factors, that could just make it, we're just going to make a lot of RNA. And it's just going to overwhelm every one of those steps. Like that, okay? So that's what's going to happen, because it turns out that when MIC is cancerous, and then we have to stop, I see, we have to stop, we're good. It says generally MIC overexpression does not stem from point mutations in the gene, and they're speaking wrongly there. They mean in the transcription unit. The MIC transcription unit doesn't change. What's going to change is the regulatory region. And there's a variety of ways that we're going to see the regulatory region can change and just make too much MIC. And you kind of flood the system. You just overwhelm the regulation, yep?
Dave, you talked about how MIC mutations are usually in the cis-regulatory region. Could we also consider like mutations in ELK or other transcription factors that would
Sure.
then have the same result?
Mm-hmm. Yeah, and so you'll see that that's going to be our focus now. What goes wrong with MIC? And the answer is going to be the regulatory region just says make a ton, and it's just like a fire hose of MIC, and that overwhelms it. The elongation, the RNA stability, the translation, the protein stability, it just pushes it too much to do that code. So overexpression is mainly due to the gene application, chromosomal application. So there's really 3 different stories of how MIC gets made too much, 3 different kinds of mutations. And so on Tuesday, we'll start with that. And so that's our last slide for today. Our 3 examples of making too much MIC. And they all 3 involve something about the regulatory region. So they're going to be really familiar to us and really satisfying. And they're just too much, right? So oncogenic retroviruses, that's one, because that's where they discovered it. What was wrong there? Then chromosomal translocation, it's in specific cancers, and then gene application, also specific cancers. So 3 concepts that we'll deal with on Tuesday. And we have a quiz at the end of class on Tuesday. You have a homework assignment that's part of that story, right? The homework assignment focuses on just a portion of the video. The quiz is on the whole video. Yep?